Okay, this is the G protein pathway, and to start it off, you have a G protein linked receptor, a G protein, and an enzyme. Firstly, reception occurs when a single molecule, in this case it would be a ligand, binds with the G protein linked receptor, therefore changing its shape and activating it. So it would change its shape to where now the G protein can bind with the G protein linked receptor, but this isn't activated yet. In order to activate this, we have to change GDP to GTP through phosphorylation. This would all be considered transduction, and transduction is the changing of the G protein to translate the signal into something that the cell can actually understand. Next, the G protein would diffuse throughout the membrane to bind with the enzyme over here. And after it binds, it would not translate. It would inform this enzyme of the translated signal, and after it does that, the enzyme would continue and do what it needs to do for the cell, because this is all for translating the message from over here to the enzyme. And then after that, the G protein would revert back to its original form over here. But since it used its GTP, it has GDP, and that would all be considered the response phase. Next is the tyrosine kinase pathway. In an attempt to prevent tissue death and failure in the cell, it does this, which is involved with repairs and emergencies and growth when needed. Each of these proteins have the ability to bind with the ligands. These have the ability to bind with the ligands outside of the cell, as well as having several tyrosine on the opposing side of each protein. After these ligands bind to these proteins, they become attracted to each other and they get closer. As if they were attracted to each other. After that happens, the proteins become active, which pulls the phosphate from the 6-ATP to the tyrosine on the other end of the protein. And in doing so, it attracts these proteins to bind with the tyrosine sites due to them being active from the binding of the phosphate and the tyrosine. In this case, the response would be the attraction of several proteins to bind with the phosphates. And now that they are being used up, the 6-ATP becomes 6-ADP. And that is the tyrosine kinase pathway. Okay, so next is the ion channel receptors. Sometimes they're also referred to as ligand-gated ion channels, I believe. But this allows sodium ions, they're not in the right spot. This allows sodium ions to cross through the phospholipid bilayer through a channel protein, which is right here, through active transport. But this is only able to happen after this ligand binds with this channel protein and opens it so these can pass through. And after these pass through, this ligand is told to move away so no more sodium ions can go through, because this is only open temporarily. Okay, so next are the intracellular receptors. These are primarily used when receiving hormones or steroids. And to begin with this process, the steroid will diffuse through the phospholipid bilayer to an unactivated protein. In this case, it would be BP. And then something called transcription factors occurs, which is when the now activated protein and the steroid they enter the nucleus, which has DNA, and it converts the DNA into messenger RNA, also known as mRNA, and after that, it leaves the nucleus. However, since this is no longer in the nucleus, it is translated into a protein to help the cell with whatever it needs to do inside of the cell. Okay, up next are the secondary messengers. Secondary messengers are relay and ion-based molecules that can freely roam throughout the inside of the cell, which is also known as the cytoplasm. To begin, cyclic AMP, also known as CAMP, is made out of adenylyl cyclase and is bound within the phospholipid bilayer. First off, the ligand binds with the G protein receptor. I'm not sure if you can see that, but it says G protein receptor, which then activates. And after the G protein binds to the receptor, even though it's not active yet, you must convert GDP to GTP, so it is now active through phosphorylation. After which, it diffuses throughout the phospholipid bilayer to adenylyl cyclase, which finally converts ATP to CAMP, which acts as a secondary messenger, which then proceeds to convert it to AMP. Okay, so this is the last part. It is an example of a secondary messenger in action. This process includes the G protein pathway, but it has a couple tweaks. And to start it off, just like the G protein pathway, the ligand binds to the G protein receptor, changes its shape so that the G protein can bind with it. However, again, it's not active yet, so through phosphorylation, GDP becomes GTP. And now that the G protein is active, it travels to PIP2, which is not active yet. And in order to activate it, the G protein must break it down into something called N-acetyl triphosphate, which is also known as IP3. And after IP3 is created, it travels back through the endoplasmic reticulum. That's not supposed to be there yet. Sorry. Back through the endoplasmic reticulum, changing its shape, as well as producing calmodulin. Sorry, not calmodulin. Calmodulin, which is calcium ions, but it has 2 instead of just 1. And finally, IP3 and calmodulin, they are the 2 secondary messengers of this process.